So I think that's pretty much everyone who's joined. Might be a few more people, but we'll make a start. So welcome to this talk. I'm Lauren and I'm just sort of introducing everybody. So this is part of our Research Your Path series of talks from young academics, which is all about research, different areas of research. And it's to sort of try and give you a taste of what's out there, what sort of stuff there is to do, and hopefully get you a bit more interested and a bit more clued up on how you might go about getting into research yourself. So first of all, I'll introduce the people who are actually going to give today's talk. So there's Dr. Catherine Hentages, and she is a reader at the University of Manchester. She came after her postdoctoral fellowship, and her work focuses on developmental biology, particularly around cardiovascular development and the genetics that sort of govern all of this. And also like lots of research over her career with that. And we also have Asta, who's one of our committee members and is a third year biomedical student. This is an area that she's particularly interested in herself. And together they've put together this talk. If you have any questions at any point, feel free to drop them in the chat, and then we'll do a sort of bit of Q&A at the end, and I'll be keeping an eye on all the things you ask and just prompt me if I miss something. So I'll hand over.
Thank you. I'll just share my screen.
You want to share screen, Hasta?
Yeah.
Yeah, just do a green button at the bottom that says share screen.
Sorry.
No worries.
Can you see that? Is that okay? I think you're on mute, Colin.
Yeah, it's all visible.
So yeah, I'll be talking about congenital heart defects and the genetic pathways that are associated with such defects. Congenital heart defects, also known as anomalies or diseases, are generally defects in the structure of the heart that are present at birth. These can be present in the vessels, the valves, and the walls in the septum. About 36,000 children are affected every year in the US, and it's the most common type of birth defect, and it's a leading non-infectious cause of death. A large number of varieties of CHDs exist, which involve right-sided lesions, left-sided lesions, corner-truncal defects, laterality defects, and isolated septal defects, which include atrial and ventral septal defects. These can be, like I mentioned, in the walls of the heart and in the septum. I'll just be quickly going over the symptoms and the causes. The most important causes are genetic and chromosomal aberrations, with other things like health conditions of the mother, medicines taken by pregnant women, lifestyle factors, and environmental teratogens also playing an important role. The symptoms generally involve cyanosis, rapid heartbeat, tiredness and fatigue, fainting, and shortness of breath, as expected if the blood is not pumped properly. Various tests involve fetal ECG, pulse oximetry that can be done after birth, chest X-ray, echocardiogram, MRI, and treatments can involve procedures during catheterizations, heart transplantations, and open heart surgeries, and medications. Several times, antibiotics are also given to prevent infection before the surgeries, and such patients are kept under lifelong monitoring. Such studies into congenital heart defects are extremely important because about 400 genes exist that are associated with these pathogenesis. These genes code for a large number of transcription factors and cell signaling transducers, which are all important for differentiation and the formation of the heart. These are all interconnected to each other and they form a broad network, but about 60% of these diseases are still unexplained because of the large heterogeneity that exists within congenital heart defects and in humans. This causes variable expressivity and variable penetrance, and it shows non-Mendelian inheritance patterns, which makes it very complex to study. These studies are therefore conducted using targeted whole exome or whole genome sequencing, trio studies, familial, cohort, and mouse models, and also patient-derived induced pluripotent stem cells. The To study the congenital heart defects, it's very important for us to understand morphogenesis of the and development of the heart. The heart is one of the first organs to develop during embryogenesis, and it is derived from the mesoderm. This involves migration of the precardiac cells from the primitive streak and assembly at the myocardial plate. The cardiac crescents then fuse together, and this forms the basis of the heart. This then undergoes right side looping, and this is important for arrangement and formation of the ventricles and the atria in the future. This further undergoes septation and then undergoes development of the conduction system and the coronary vasculature. A large number of genes are involved in each of these steps, and therefore mutations in any of these genes can cause tremendous defects. And the most important thing about the heart is that it is the left-right symmetry of the heart is very important for oxygenation of the blood and for pumping of the blood. The genetic causes are broadly classified into single gene mutations, which can be with or associated with or without syndromes. Aneuploidy and microdeletions, single nucleotide polymorphisms or SNPs, and copy number variations are also associated with congenital heart defects. A large number of transcription factors are generally involved in cardiogenesis. The most important ones being GATA4, NKX2-5, and TBX5. And I'll be talking about a few of these today. So beginning with a single gene defect associated with syndromes, the most common and the most important one being Holt-Oram syndrome causative mutation in TBX5. So Holt-Oram syndrome is an autosomal dominant disorder which is caused which causes upper limb malformations and congenital heart defects like atrial and ventricular septal defects. This is caused due to mutation in TBX5 gene, which is a part of the TBX transcription factor family. A large number of frame shift mutations, nonsense, and splice site mutations can cause such defects, and which it prevents it from interacting with NKX2-5 and GATA4, which prevents formation of the conduction system and development of the heart. TBX5, as mentioned here, contains, so TBX5 is basically just one of the transcription box factor, and a large number of them are involved in the formation of the heart. TBX5, however, is involved in chamber formation, septation, and cardiomyocyte differentiation, and mutations can cause loss of chamber, myocardium, and looping defects. It serves as a transcription activator for a large number of genes, and it also interacts with a large number of genes, the most common ones being NKX2-5 and GATA4. As seen here, the I and L images show the wild type hearts of a mouse, while J, K, M, and N show the mutated versions. The red arrow shows complete formation of the atrium, while the red arrow and the red asterisk in J and K show atrial septal defects. M and N, the asterisk and the arrow again show ventricular septal defects. TBX5 is mainly associated with the first heart field, and overexpression of this causes loss of ventricular septum, while mosaic expression of this causes formation of an additional ventricular septal. Mice that are homozygous for this null mutation can establish a linear heart tube, but it does not allow the formation of the first heart field. And as mentioned, in humans, this causes Holt-Oram syndrome. Retinoic acid signaling restricts TBX5 expression in the posterior part of the heart tube, and STAT3 signaling activates the expression. And I'm not sure if you can see my arrow, but that's what I'm pointing to. TBX5 also acts as a transcriptional activator, as mentioned, and it interacts with TIP60, which is present in the cardiac muscles. Trisomy 21, also known as Down syndrome, is associated with aneuploidy and microdeletions. So that is chromosome 21 that is generally associated with Down syndrome. And a large number of genes are associated on this chromosome that can cause Down syndrome, and congenital heart defects associated with it. And about 40 to 60% of such individuals have congenital heart defects, which can involve tetralogy of Fallot and atrioventricular septal defects and other things. So due to the presence of an additional chromosome 21, the TSCAM gene is multiplied, which increases cell adhesion in the endocardium and causes atrioventricular septal defects, while collagen 6 is involved in the formation of the atrioventricular septum, and also overexpression again causes the atrioventricular septal defects. KCNJ6 is a gene which codes for G protein coupled potassium channels, and overexpression can cause arrhythmogenicity. While RCAN1 encodes a calcineurin inhibitor, which I'll be mentioning later. So again, these are the various defects that can be caused. So in the study, a transchromosomic mice was generated which contained HSA21, which is a similar gene to chromosome 21 in humans. And it shows ventricular septal defects with E being the wild type version. And F shows membranous ventricular septal defect, while G, the arrow here, shows muscular ventricular septal defect, and H shows the same from the top. It also shows a large number of outflow tract defects. And again, A, B, and C are the wild types, while D, E, and F are the transchromosomal mice. The pulmonary artery, as shown by the white arrow in A, arises from the right ventricle, and the aorta arises from the left ventricle. But in the transchromosomic mice, the position of the aortic valve, as shown by this white circle here, is shifted rightwards. And E and F also shows double outlet right ventricle, where the pulmonary artery and the aorta both arise from the right ventricle. Defects can also involve AV canal defects, which, as shown in the wild type hearts, the black asterisk in A shows that the AV junctions are separated by the AV septum, and the light on the tricuspid valve and the mitral valves are shown by the black arrows here in B. However, in case of E and F, these are fused together, and there is no wedge-shaped structure that is seen in A and D. As mentioned, RCN1 is responsible for the calcineurin NF-AT signaling pathway, and in this case, stimulation causes increase in calcium levels in the cell, which binds to calcineurin, which is a calmodulin-dependent serine 3-unit kinase. This causes dephosphorylation of NF-AT, which will migrate from the cytoplasm into the nucleus. It will bind to GATA4 and will cause activation of transcriptional factors and target genes, including DSCR1, which is also known as RCN1, which I mentioned. This increase in the level of DSCR1 causes inhibition of calcineurin, which will inhibit this pathway. As mentioned, in case of trisomy 21 or Down syndrome, there's an increase in the level of DSCR1 due to presence of a third copy of chromosome 21. This causes increase in the suppression of calcineurin and causes suppression of the target genes and prevents further formation of the heart. And KX2-5 is a single gene defect associated with non-syndromic cardiac malformation. It is important for a large number of factors during cardiac morphogenesis, which involves looping morphogenesis, chamber septation, and chamber specification. And it's also important for the formation of the conduction system and homeostasis later on in adults. It also regulates the expression of a large number of cardiac genes and transcriptional factors, including HAND1 for the development of the left ventricle, BMP2 for the regulation and differentiation of cardiac outflow tract, and BMP10 for cardiac trabeculation development. This is the gene which and mutations in these genes can cause a large number of defects. And 2 of these mutations prevent NTX2-5 to bind to the DNA and causes haploinsufficiency, while the 3rd mutation enhances DNA binding. Heterozygous mutation in NTX2-5 causes AV block and atrioventricular septal defect due to decrease in nuclear import, downregulation of BMP and NOT signaling pathways, and dysregulation of the genes which are involved in this cardiomyocyte differentiation. So these are generally the cause of the sort of defects that are caused due to mutation in NTX2-5. A and B show the wild type versions, while C, D, and E show the mutated versions. C shows a membranous ventricular septal defect, while D shows muscular ventricular septal defect. So I'd also like to bring about this concept of polymorphism that exists in case of congenital heart defects. And heterogeneous knockout mice in inbred strain with a background of C57 with the background of C57 strains generally have atrial and ventricular septal defects, while strains FBB and AJ generally have susceptibility for atrial and ventricular septal defects. So in a particular study, animals were backcrossed with C57 for several generations to create a pure knockout mouse with NKX5 mutation having a C57 background. These males were then crossed with females having of FBB strain or AJ strain to generate the first progeny, the F1 progeny. And the F1 progeny were then intercrossed or backcrossed with the parental strains to cause the F2 progeny. So a large number of studies were then done in this case, and it showed that the wild type versions had atrial and ventricular septal defects while this decreased in the first progeny. In the second progeny, however, atrial septal defects were higher in the crosses that were with AJ, but they were lower in the crosses with FJ in the intercrosses. Interventricular septal defects that showed a significant increase in the crosses with AJ, but not with FJ with the strains. And there was not much difference in the ventricular septal defects. And it shows that the AJ strain basically had polymorphism that enhances its susceptibility to atrioventricular septal defects and atrial septal defects. It also showed that membranous and muscular ventral septal defects were also found in such hearts, but these were common in the C57 background, and the incidence of membranous and muscular ventral septal defects were associated with the amount of C57 alleles that were present, where muscular septal defects increased with the amount of C57 allele, while membranous ventricular septal defects decreased. As mentioned, a large number of transcription factors and signaling molecules are important for the development of the heart. And this is just a partial list that is shown here. This brings about the concept of canalization, which involves development that flows through a series of channels to produce an invariant wild form. Here, important phenotypes are maintained at a stable optimum, while large mutations tend to decanalize the individual, and cryptic invariants of these modifier genes will be expressed, which do not affect the wild types. This increase in complexity of this congenital heart defects, this increases the complexity of congenital heart defects. Protective polymorphisms, however, exist against certain mutations in humans, which tend to serve as a buffer against these harmful mutations. As seen earlier, the F1 progeny did not show defects due to heterogeneity and a near complete suppression of the harmful phenotypes, while homozygosity at some loci in F2 contributes to manifestation of a certain defect. So a large number of genes are important for the development of the heart and can influence the development of various types of congenital heart defects. And we're just beginning to understand these genetic influences of such defects, especially in ethnically diverse populations. And thus more knowledge is needed on the genetics of these to develop treatments and providing genetic counseling and care to the affected individuals. And yeah, that's all. Sorry, I can just stop sharing my screen. So Kathy, you can share yours. Would you like me to share your screen?
Sorry, so do you want me to move on to do my presentation, or do you want to take questions yourself first?
I think we can take the questions at the end together.
That sounds fine. Let me just share the right window then. Okay, I hope you can all see my window then. So I'll begin my talk following on from what Asta's very nicely explained about the studies and the reasons why we studied the genetics behind congenital heart defects. I'd like to give you a talk on the research we're doing in my lab and a bit about the research strategies we use to try and identify genes that are associated with congenital heart defects. One thing that we do is a particular type of experiment, which is the mouse mutagenesis screen, and that's been the basis for all the research that's going on in my laboratory at the moment. So as Asta very nicely introduced, congenital heart defects are quite a large problem. They are present in about 1% of live births, and probably also are the causes behind rather a larger number of stillbirths and miscarriages. And although we know how to now diagnose and identify many of the different types of congenital heart defects, really understanding the genetic basis to them is still a challenge. So in many cases, especially in many families, the specific genes that cause a congenital heart defect in an individual are unknown. And the way that my lab has tried to tackle this problem is to take a forward genetic approach. This means that we don't start by selecting a gene, but instead we look at phenotypes. So what we do is we use a chemical called ENU, and that generates random point mutations in the mouse genome. We then screen animals to identify those that have cardiac developmental defects that model the defects we see in human congenital heart defect patients. And by doing that, we then can identify genes that were not previously known to have a contribution to this disease. So there are some challenges to undertaking this approach. One of them is that once you have identified an interesting phenotype that models a congenital heart defect, you then still have to identify what gene has been mutated. And that can be quite challenging. I'll talk about in the talk today about how we've approached that in my lab. Also, when you identify a new gene that contributes to congenital heart defects, it can be difficult to understand why that specific gene does play a role in causing heart defects. And I think the gene I'll tell you about today is a good example of that challenge. But there are benefits to this approach as well. So we reduce investigator bias because we're not selecting the gene at the outset of the study. We're just saying we want to look at phenotypes that we know are similar to human disease phenotypes. And therefore, we're not biasing it towards valuing the importance of any particular gene in our study. This approach also generates hypomorphic alleles that are caused by point mutations, and that's very similar to what happens in human disease states. So rather than the mouse knockout alleles where a whole gene is deleted from the genome, we're creating point mutations, and that's more similar to what happens in humans where they acquire new point mutations in genes of interest. And because of this, we can identify new genetic links between genes that were not previously likely to be associated with a particular process. And I think, again, the gene I'll tell you about today is a good example of how we've identified a new genetic link between a gene and congenital heart defects. So the mouse mutant that we're studying is called L27, or lethal 27. These are some examples of heterozygous embryos from this cross, which are essentially wild type in their phenotypes. You can see here, this is a mouse embryo. We have the developing head region here. This is developing tail. And this structure right here is the developing heart. And this is just a view of the embryo from the other side. As Esther mentioned, one of the events in heart development that occurs is called looping of the heart. And that looping happens in a specific direction to set up the left and right side of the heart so that they take on the different identity and the different connections to the great vessels that are required to form vascular connections with the lungs and the rest of the body to ensure that you have the correct movement of blood throughout the organism. And so this is just showing you both views of the heart. And this is an embryo itself in a structure called the yolk sac. The yolk sac is a developmental membrane structure which undergoes remodeling to form the vasculature. And it's important for having the vascular connections between the embryo and the placenta and the mother. And if we look at our mutant embryos here, one of the things I hope you can note is that they have distended hearts. So this here on the mutant embryo is the developing heart. And here's the head region. You can see here that in our control embryos, the heart is much smaller than the head. In our mutants, the heart is approximately the same size as the whole developing head region, indicating that the heart tube is distended during development. This may be an indication of abnormal function. And also, again, here's our yolk sac structure. And we can see that this looks very bumpy. You can see here that we have some blood in the yolk sac, but these blood vessels of the yolk sac have not rearranged to make large, long vessels as they do in the control. So here's some large, long vessels in the yolk sac of the control embryo. So this again indicates a compromise of the cardiovascular system in these embryos. And these embryos are embryonic lethal, then they die at mid-gestation. One of the things that we noticed was that these embryos have cardiac looping defects. So this is a front-on view of the embryo. Again, this is our head region, and the top panel is all control embryos. The bottom panel is our mutants. And you can see here, this is the heart tube. This circular structure here is the developing presumptive ventricle, and it's looping around the embryo. So this is a leftward looping. In our mutant embryos, we see that the heart has undergone looping, but it's looping in the reverse direction. So again, this looping of the heart, this presumptive ventricle is now going to be forming on the opposite side of the embryo to where it should. So this indicates a left-right reversal of the developing heart. And these are some images here showing an experiment called in situ hybridization, which is used to identify the gene expression pattern of a gene called NKX 2.5. It's one of the genes that Asta told you about that's very important for heart development. It's expressed in the heart tube. And we're just using it here as a way to indicate the heart tube in these embryos. And you can see if you compare the images from the top and bottom, the structure that you can see highlighted in this purple color is quite different. And this is, again, because the hearts are looping in the opposite direction in our mutants as they do to the controls, indicating a reversal of the left-right direction of the heart tube. And we see when we analyze these embryos slightly before mid-gestation that about 50% of the mutants show a reversal in the heart looping direction. So the looping direction of the heart is one of the first readouts of a process that happens early in embryonic development, which is the establishment of the left-right axis. And this axis is established by signals that occur at a structure in the early embryo called the node. So the node is a structure at the midline of the embryo. So this is a developing embryo here showing you the head and the tail region. And then there's a structure here kind of in the middle of the embryo called the node. And the node is a pit of cells. So this again is showing you, this is a 3D image of a mouse embryo here showing you the node, which is this little invagination here, midline of the embryo. This is the developing head region of the embryo here. And the node is a pit of cells that are ciliated. And these cilia on the node cells are motile. And the movement of these cilia creates a net fluid flow to the left-hand side of the embryo. So this is showing you the embryo here with the right and left sides. And the movement of the cilia then creates flow that goes to the left-hand side of the node. This results in a change in gene expression patterns around the node, most notably of genes called nodal and lefty2. And it also then results in a change in gene expression patterns on the left-hand side of the embryo along the length of the embryo in a particular tissue type called the lateral plate mesoderm. And so therefore the readout of the function of cilia at the node is that you can see the change in fluid flow on the left. So of course, because we noticed that these embryos had this change in heart looping direction, we then wanted to look at the node structure and node cilia and determine whether that had been altered in the mutants. So what we have here is, again, some embryos at different stages of development. This is a heterozygous embryo, essentially a control. And this is our mutant at a corresponding stage of development. This is the node structure here. And this is a higher magnification view of that structure. And one thing that we noticed is that the node morphology is slightly altered. So the nodes seem to be more flat in our mutants. Again, if we look slightly later in development, you can see here that the node is very much a pit in our control embryo, and it again remains quite flat in our mutants. But one thing you can also probably notice by these little white hair-like structures here is that we do have cilia projecting in both the controls and in the mutants. Let's change the slide. No, I can't seem to move it forward at the moment. There we go. So this is a higher magnification view, just showing you the cilia at the node. Once again, these are control embryos here on the left and our mutants on the right. And again, you can see these white hair-like lengths here are our cilia. So each of these white strips is a cilium on a cell at the node. And so we can see that clearly cilia are present in our mutants. We've also used antibody stains to identify these cilia using green fluorescence here in these images. And these are looking at the node sort of flat on. You can see that the node in the mutants is maybe slightly misshapen compared to the controls. And again, this is a side-on view of the cilia. You can see that in the controls, these cilia project upwards into that pit that's created by the morphological structure of the node. In our mutants, the green cilia here are shorter and they are not kind of projecting as much into the cavity that's created by the structure of the node, because as I said, in the mutants, the nodes are much more flat. And so there isn't as much of a pit there for these cilia to project into. My PhD student who did this work then painstakingly counted the number of cilia in the nodes of all of our mutants, and also the length. He measured the length of the cilia in our mutants. He found that there were no significant differences in the number of cilia at either stage in the mutants and controls. And I think that's probably, you can see that from the images as well. There don't seem to be drastic differences in the numbers of the sort of white hair-like structures that you could see. However, when he measured the cilia, he did find a statistically significant difference in the length of the cilia, and in particular at the later developmental stage. In the mutant, the cilia are shorter, significantly shorter than they are in controls. Now, in experiments done by physicists who have modeled how the cilia movement creates fluid flow, they know that the amount of flow created is proportional to the length of the cilium. So a shorter cilium will produce a less forceful flow. And so we can interpret this research finding as that probably the mutants have a weaker flow at the node, which might then impact on the gene expression patterns that we could see. We wanted to ask whether motility could be affected in a structural sense in the cilia in these mutants as well. So we did some transmission electron microscopy, where we could see then the structure of microtubules present in the cilium. And these microtubules are motor proteins that are needed for the cilium to have movement. We've looked at that in the controls and in our mutants. And we see no difference in the amount of the motor proteins that we see present in the structure in the cilia. So we've looked at the control wild type, a heterozygous, which is essentially a wild type phenotype, and our mutants. And we see no differences in the ultrastructure of the cilia at the node. We then measured motility. So we created high power movies of the cilia beating at the node. Unfortunately, can't play the movies for you today as part of this presentation. I couldn't make the movie files work, but I'm able to just show you kind of a still from them. And you can see here all these little white lines in our wild type control are moving cilia that are captured at a particular phase of their movement. And you can see that we don't see those same kind of lines in our mutants. We see very small, shorter cilia, as these kind of little white dots indicate, and they were largely non-motile. So we counted, again, the number of moving cilia we could see in the nodes of each of the different genotypes that we filmed. So our wild types and heterozygous embryos had motile cilia, and there was no significant difference between the numbers of motile cilia. But when we compare our mutants, shown here in gray, to the wild type numbers, we get a significant difference. And effectively, we had only, I think, one motile cilia detected in all of the mutant embryos that we analyzed. So we did not see any cilia motility in our mutants. We can ask then, how does that loss of motility impact upon the flow of fluid around the node? And we measured that by placing beads on top of the nodes in these embryos and tracking the movement of these beads across the node. We can see here in a heterozygous embryo with a normal phenotype that we have a movement towards the left. So this is the right side of the node, this is the left side of the node, and these lines represent the track of a particle. And so this is 3 different particles that we tracked, and they all have a net movement to the left-hand side. In our mutant embryos, again, we have tracked the movement of particles, of beads on top of them. And you can see here that they're much more static. Although the beads do bounce around a bit due to Brownian motion, we don't really have a net generation of movement to the left-hand side of the embryo. So we can see that the shortening of the cilia and the lack of motility of the cilia results in a reduced fluid flow in the nodes of these embryos. We have no flow progressing towards the left-hand side. If I can move on from this slide. Okay, and so as you might expect then, we would predict that we should see a difference in the readout of the left-right axis formation. So we would expect that all those genes that get expressed in the left-hand side of the embryos when you have normal fluid flow at the node would not be expressed in our mutants. And that is indeed what we see. So we've looked at embryos here. Again, these are heterozygous control embryos. In this case, we're looking at the head of the embryos at the top of each image, the tail region of the embryos at the bottom of the image. Our node structure is here approximately at the middle of the embryo. And I've got right and left labeled at the top of each image. And what we'd expect, and what we do see in the controls, is that we get gene expression patterns more prominently on the left-hand side of the embryo. And this is for several genes involved in the laterality process, genes I mentioned before called nodal, lefty1 and 2, and PIDX2. And what we see in our mutants is a disruption of that gene expression pattern. So if you compare a mutant to the corresponding control, you can see that in the control we have leftward gene expression, and in our mutants we lose this gene expression on the left side of the embryo. And that is therefore a likely result of the loss of the flow at the node of the embryos due to the abnormalities we see in the cilia at the node. And so that kind of explains why our heart development, the laterality direction of our heart development would be randomized then in the mutant. So we don't have proper expression of these genes that are needed for setting up the left-right axis and telling the embryo the positional identity of its organs, so whether that should be on the left or right side of the embryo. And so we've disrupted that process because we've lost the cilia function at the node quite early in development, and that's having a readout in abnormal heart formation. Now, this abnormality in cardiac formation is actually similar to a human disease condition, which is a set of diseases called heterotaxy. And one way that we could ask whether our embryos are really a model for heterotaxy is to ask, does the heart developmental direction agree with the placement of other organs? So in humans with heterotaxy, they have a mismatch of the left-right orientation of their organs, and that results in some quite complex developmental abnormalities and quite complicated compromise of cardiovascular function because they have leftward placement of some organs, rightward placement of other organs, and that creates kind of a mismatch in the overall connection of the organs in the body. And so because our embryos are lethal during development, it's difficult to really truly analyze all of their organ placements. But one thing that we were able to do was analyze the placement of the developing stomach. So we looked at the expression of a gene called BARX1, which marks where the developing stomach is in the embryo. And what I've shown you here is a heterozygous embryo and a mutant. I've identified the midline based on the neural tube position of the embryo, and that's shown here in this purple dashed line. And we've outlined the heart looping direction in red here. And what you can see in purple is the expression of BARX1, which is a marker of the developing stomach. And you can see here that our control embryo has correct looping of the heart. So here's the development of our presumptive ventricle. And the presumptive ventricle and the developing stomach are on the same side of the embryo. Okay? In our mutants, we have the reversed heart looping direction, but those embryos with reversed heart still have the stomach in the correct place. So this indicates that these mutants have a mismatch between the heart looping direction and the placement of the stomach. And again, that would indicate that they have heterotaxy or serve as a model for this more complicated congenital malformation where the organ direction is actually not in register with each other. We have some organs taking on a leftward position and some organs taking on a rightward position. Okay. So as I mentioned earlier in the talk, one of the challenges of using this approach of doing mutagenesis is that although we have characterized the mutants, we know they have a very interesting phenotype that will probably reveal things about the genetic processes involved in cardiovascular development. We also, because of the strategy we took where we create random point mutations, we don't know which specific gene has been mutated to cause this very interesting phenotype. So we used a set of experiments called meiotic mapping, where we were able to reduce the candidate region for this phenotype down to approximately 110 genes in the genome. So it's a region of mouse chromosome 11 that contained 110 genes. We did whole genome sequencing on homozygous mutant DNA samples. We identified from the sequencing 35 different sequence variants that were present in a large percentage of the reads. But of those 35 variants, only one affected a coding region of a gene. And since these mutants have a quite severe phenotype, we would expect that we have altered the coding region of a gene with this mutation. And that one coding region variant that we found was a non-synonymous amino acid change in a gene called PRPFA. So we went on to confirm that these animals had indeed acquired a mutation in the PRPFA gene. We did this by looking at an additional 3 different mutant embryos, and in all 3 embryos, we could confirm this homozygous sequence change. We looked at heterozygous embryos, again, an additional 3, and confirmed we could see that they were heterozygous for the base change at this position in the PRPFA gene. And we could confirm that that base change was not present in either of the 2 strains that were involved in the mutagenesis screen. So this change changes an A to a G in the genome, and this is found in our mutants, but not in any of the wild-type mouse strains. When we look at what this sequence change does to the protein coding reading frame, we see that it creates a change in a conserved amino acid. It changes an asparagine to a serine, and this particular amino acid is conserved in several different species. We've looked from mouse to yeast. It's also conserved in human. And so that probably indicates that this amino acid change will have a functional consequence in the protein, because it's a very conserved amino acid and it's conserved highly throughout evolution. That would indicate probably an important function for that amino acid. And it's in a region of the protein that's very highly conserved as well, indicating that that region of the protein probably has an important function. When we look at where that amino acid change actually is, it's shown here in purple in the structural model of the protein. This region of the PRPFA protein interacts with another protein called AAR2, and so it's very likely that our missense change changes an amino acid that's at this interface of the protein-protein interaction, and then therefore could destabilize this protein-protein interaction. So that's interesting, but one thing, of course, the immediate thing to wonder is, well, what is the PRPFA gene? And what is it known to do? Has it been identified in human congenital heart defects previously? So PRPF8 stands for pre-mRNA processing splicing factor 8. It's a spliceosome protein. It's a component of both the major and minor spliceosomes. It's the most highly conserved nuclear protein with 60% amino acid identity from yeast to human. And it's central to the expression of all nuclear intron containing mRNAs because it's critical to the splicing process. PRPF8 within the spliceosome contacts the intron that is being spliced out of the transcript. And so the role of this protein is clearly very critical to completing a splicing reaction. Now, as I said, our particular mutation that we see is an amino acid residue that's conserved from all the way from yeast to human. And of course, we wanted to know, has human PRPFA been identified in other diseases? Is it associated with congenital heart defects? One thing when we began to research this gene, we discovered was that the gene is associated with autosomal dominant retinitis pigmentosa. And this is associated with mutations that occur in the final exon, so at the very C-terminal portion of the protein. And those variants disrupt the interactions between PRPF8 and another protein called BER2. And this has been characterized in studies of retinitis pigmentosa with quite a lot of research really demonstrating that these protein-protein interactions are distorted by these retinitis pigmentosa mutations. Knockin mice have been created for one of the human retinitis pigmentosa mutations, and the adult mice were analyzed. These homozygotes are viable, and as you would expect for a model of retinitis pigmentosa, they have defects in their retinal structure and function, but they clearly do not have the severe cardiovascular defects that we see in our PRPFA mutant mice. And this discordance between the phenotypes is most likely due to the fact that we have a different amino acid change in a different region of the protein. So our amino acid change is in this region of the protein, more towards the middle of the PRPFA protein, which, as I said, mediates interactions with another protein called AR2, and therefore may affect PRPFA function in a different way to these retinitis pigmentosa mutations. And also I should say that these retinitis pigmentosa mutations are in humans occurring in a heterozygous state, although in mouse you can make animals that are homozygous for these retinitis pigmentosa variants, and they are viable in the mouse. So we then looked at the expression pattern of PRPFA in mouse embryos, and this is just in wild type embryos, because we were kind of intrigued as to why should a protein that's quite specific in its phenotype that it produces in terms of our congenital heart defects that we see, why would a protein that's involved in such a general process as splicing be causing that specific phenotype? And so we looked at the expression pattern, and we see that it's more dynamic than predicted. We would think that PRPFA must be expressed in every cell, and actually I'm sure it is at some level. But we can see that we have in early development much more intense regions of PRPFA expression, shown by this dark purple color here, on regions of the embryo that are affected in our embryo. So this staining here is around the mouse node. And so we know that the node and the left-right axis are affected in our embryos, they have abnormal phenotypes, and we see more expression of the PRPFA around the node. We also see it in the head region, which we also find abnormalities in our mutant embryos, and along the neural tube and in structures called the somites, which also are often abnormal in our mutant embryos. So it seems like tissues that are more highly affected in our mutants have higher expression of PRPFA, which could indicate a greater demand for its function in those particular tissues. But what I wanted to kind of convey to you today is that by beginning this project looking at congenital heart defects, and we're looking at in particular the left-right axis formation and defects called heterotaxy, we actually think that our research now can have insights into the disease of retinitis pigmentosa as well. And it's known that PRPFA as well as other splicing factors cause retinitis pigmentosa. The mechanism for why, again, these splicing factors cause these very specific adult onset eye defects is unknown. And I should say that retinitis pigmentosa is an adult onset disease. It's characterized by progressive vision loss, especially night vision can become affected. And that happens often as individuals are reaching their 40s and 50s. So it's clearly not necessarily a developmental disease. And really, it's unknown why splicing factors can cause retinitis pigmentosa. It's presumed that there may be high metabolism in photoreceptor cells, and that this high metabolic activity in these photoreceptor cells just requires a large amount of splicing in order to cope with the protein turnover that they need. But what we think is very intriguing and is a nice link from our studies as well is that the photoreceptor outer segment of the photoreceptor cell is a modified cilium. And clearly we know that there are cilia links with PRPFA from our own studies on our mouse mutant, but collaborators of ours at Leeds University also showed that if you knock down the PRPFA gene in ciliated cells, and this is research done in cell lines, you lose the cilium on those ciliated cells. So this is a control cell, and you can see in red here, the cilium. This is again an example here in red of the cilium on these cells. And if you knock down PRPFA, you lose the formation of that cilium. Okay? So that again indicates that PRPFA may have some function specifically in the cilium, or at least may have a particularly high requirement in the formation of the cilium. And that may give us some insights into the causes of retinitis pigmentosa and why splicing factors are associated with retinitis pigmentosa, as well as the heterotaxy defects we see in our mutant mouse. We have identified ourselves that PRPFA protein is expressed at the cilium, and I should note that the collaborators in Leeds have identified this in cell lines. We looked at it in our mouse mutants. We can see PRPFA protein here in red, and in green we see the structure of the cilium, and we can see if we look in the cytoplasm that we can see the association of PRPFA protein at the base of the cilium. So again, the cilium is labeled here in green. Our PRPFA protein is here in red. We also have shown that not only in the mouse, but also in zebrafish, we can generate a model for heterotaxy, where we can see that if we knock down PRPFA function in the zebrafish, we get a reduction in heart looping, and this is sort of incorrect heart looping. This model is kind of these descended heart tubes that we see in some of our mouse mutants. And so we can see that when we knock down PRPFA in zebrafish, if you look at this bar chart here, the panels in gray here showing you the percentage of total hearts analyzed that were unlooped. We see a greater proportion of unlooped hearts in our knockdown zebrafish model. So this indicates that in different species, this function of PRPFA is conserved. And so that led us to form the hypothesis that the PRPFA mutation causes missplicing of cilia genes. We generated an analogous missense mutation in yeast. Again, I said this amino acid is conserved all the way down to yeast. And yeast is a very nice system because you can do a splicing assay in yeast, where the yeast are forced to kind of perform splicing on an artificial reporter, an artificial intron, and you can get a readout of how much splicing occurs. We could see the amount of splicing here in blue with the wild type version of PRPFA. And you can see that when we make our mutant version, shown here in red, we have a reduction in the amount of splicing. Our current work focuses on looking at splicing of cilia genes in our mutant. This is one example. I'm just showing you where we discovered in our mutants that there may be a new splice site that's created. This is showing you the splicing of a wild type control embryos of a gene called IFT46, which is a gene important for cilia function. And these little patterns are just showing you where splice sites are present. And what we see here in our mutants is we have the usage of a new splice site in the mutants. And so that may indicate that we're seeing splicing in cilia-related genes at inappropriate locations in the mutant. And of course, that splicing may therefore have an effect on protein function of these genes related to cilia. So I'd just like to summarize what I've told you about our research then. We've identified this mouse mutant that has a cardiac left-right asymmetry defect. It's a model of a human congenital heart disease called heterotaxy. And we have identified certainly that there's misexpression of left-right asymmetry genes, which may be related to missplicing of cilia genes and also potentially left-right asymmetry genes that are required during development. This mutant is caused by a missense mutation in a gene called PRPFA, which is highly conserved. And this gene also is known to play a role in retinitis pigmentosa. And so we think our research not only gives us insights into congenital heart defects, but will also lead to some new understandings into generally ciliopathies and also specifically the retinitis pigmentosa disease. Okay, and with that, I'll conclude the talk about the research that's been going on in the lab. Most of what I showed you today was the work of my PhD student, Michael Boylan, with contributions from other lab members. And I should note that I have 3 members in the lab currently working on this project, Wase, Dale, and Nuf, who have all joined the call today. And unfortunately, I didn't add any of the new data that they've been generated into this presentation today, but they're currently taking this project forward. And we have, of course, collaborators in Manchester and other institutions who have been instrumental in facilitating this research. And we couldn't have done any of it without funding. So I would like to thank the British Heart Foundation, BBSRC, and Syngenta, who provided funding for various stages of this project. And with that, I will stop sharing my screen. And I'd be happy to answer any questions.
Thank you so much, Catherine. That was absolutely fantastic. And Asta at the beginning as well, really good introduction. So we can open the floor out to questions as well for another about 5 minutes or so. So if anyone has any questions, please feel free to ask. I had one question actually that I wanted to ask was, there's a lot of students here who are like really like complete novice and new to the field. And if they were to go about, so it's quite a general question, if they were to go about doing some of the experiments that you've shown, particularly like using a mouse model or using like a model that they're not really familiar with, like maybe they know the human anatomy particularly well, but they don't really know all the other models, how would they go about like educating themselves so that they have a good foundation to be able to do these experiments?
Yeah, I'd say there's lots of really good review articles out there. So if you begin kind of an introduction to the literature, often a data paper is just kind of, full of so much specific terminology that it's kind of hard to grasp. So I always recommend students start by reading some review articles, and depending on, the field and the specific area, some of the reviews are presented either for scientists of different specialties, so they're quite general, or people who are at kind of a more earlier level in their research careers. And so I would suggest looking at review articles, but also textbooks. There are not probably the general kind of textbooks that you would use in your courses, but there are some like sort of reference textbooks that are used. There is one textbook on mouse development that I always recommend to project students to start looking at. I know the Manchester Library has it. And it's a very comprehensive textbook that goes through each different organ system. And it again is aimed more at educating someone who hasn't really had a deep introduction to that topic before. And there are other reference textbooks as well. There is again a specific textbook for mouse cardiac anatomy that again is a good introductory book. And there will be these similar types of books in other organ systems in other fields as well.
Brilliant, thank you so much. I think everyone's just thanking everyone lots at the moment, nursing students, and yeah, thank you so much for the informative presentation. So I'll just put in the chat now that we're waiting as well. I think it was already sent before, but for everyone who wants a portfolio certificate from this event, if you just complete this feedback form, I'll put it in the chat again, and it will link you to a portfolio certificate at the end that you can download. Thank you so much again, Catherine and Asta. I think that is all the questions. I don't think anyone has any particular questions at the moment. And thank you for all your PhD students for coming along as well. I actually think I've got one direct question. Can a person integrate pediatric cardiac surgery with genetics?
In terms of sort of medical training, or like kind of conceptually research-wise?
Well, they've not said. I'll just ask.
Yeah, I'm not entirely sure about the routes of training and whether that's like feasible as you're progressing on your training to become, say, a registrar or something. But definitely from a research perspective, they are integrated. And one of my collaborators at the university is Professor Bernard Keaveney, who is a cardiologist, and he does kind of a range of things. So he sees patients with atrial fibrillation, and he also sees patients with congenital heart defects. And he has two kind of disparate specialties, one being adult patients with atrial fibrillation and one being childhood patients with congenital heart defects. So that diversity in what clinicians do is certainly possible, at least based on his example, but I don't know very much more about the specifics of medical training.
Actually, Dr. Keaveney did a talk for our series as well. So like that lecture is on our YouTube channel. So you might want to check that out, because he went through actually the whole like his research areas and everything and the genetics behind it. So that'd probably be more specific, like more clinical, rather than this is a bit more of the science and the laboratory side of the talk. All right, I think that's all the questions. Thank you once again, Catherine and Asta. I hope you guys have a lovely evening.
Thank you. And thank you for inviting me to do the talk today.
Thank you so much. All right, take care.